It's my pleasure now to introduce our first keynote speaker, Professor Gerard Graham, who is the head of the CHEMOKINES research group at the University of Glasgow in Scotland and deputy head of the Institute of Infection, Immunity and Inflammation, a long-time colleague and a good friend. Gerry works in the field of chemokine biology and is actually responsible for very many of the seminal discoveries in this area. His two main interests in chemokine research are that his group is amongst the world leaders in the study of what are called atypical chemokine receptors and their role in the regulation of the function of chemokines. And he's also known for using complex genomic approaches to understand the orchestration of the chemokine-driven inflammatory response. He's extremely well funded, which is delightful in this day and age of problems with funding, by the Medical Research Council program and by the Wellcome Trust, for which he is chair of the expert review group in the immune system in health and disease. Gerry is also a recipient of many awards, including the Wolfson Royal Society Merit Award, and has also been elected as a Fellow of the Royal Society of Edinburgh in Scotland. Rather than take up any more time, as I can see Gerry on the screen now, it's my pleasure, Gerry, to introduce you to give your talk, and over to you.
Can everybody hear me? That's okay. So how do I put my presentation up, Justin? That's it. Thank you very much. That's great. Well, welcome. It's a great pleasure to be able to speak to you, and I'm very grateful. And I've called the talk Finding Direction of the Inflammation Highway, and hopefully the reasons for that will become apparent as we go through this. My name, as Eleanor said, is Gerard Graham, and I'm from the University of Glasgow, so greetings from Scotland. And this is my email address here, so please, if there's anything that I say that is not clear, or even better, that you don't agree with, or if you want to ask any general questions with sort of science that I'm involved in, please contact me and I'll be delighted to engage in a discussion. Now, if you go back around 35 years in immunology, one of the main questions that was unresolved was, how do white blood cells know where they're going? So these cells have to navigate in space and time very, very precisely. So these beautiful immune cells that we have that form the adaptive immune system have to get to specific places. So in my skin, for example, I have T cells that have gone to the skin, and they've gone there precisely. They've not gone there by accident. And in my gut, which you don't want to see, I also have T cells that have gone to the gut, and the same in the lung, and the liver, and the brain. And so the idea is that these cells navigate precisely. And what we're going to be interested in this morning is how these cells, these inflammatory effector cells, do the same thing. So how do these cells get to specific tissue sites to initiate localized inflammatory responses? And that's really important because some of these cells, particularly neutrophils, are extraordinarily toxic. So you've got to get them to the right place at the right time, or you can cause, as we'll see at the end of the talk, really extremely debilitating diseases. So first of all, let's consider inflammation. The basic biology of inflammation is always shown in this sort of slide here. These are what are called the 5 cardinal or main signs of inflammation. There is heat, characterizes inflammation. The inflamed site is red and it's swollen, it's painful, and there's what we call loss of function. And that's important because you want to ensure that you don't damage the inflamed site even more by using it. So loss of function means that you rest the inflamed site. Now, many of these signs of inflammation are a simple product of the fact that one of the first things that happen in inflammation is that blood vessels start to expand and fluid pours out from the blood, and it's pushed out by the force of the blood. And that fluid enters the tissues. That causes the redness in the tissue. The fluid carries with it molecules called pyrogens, and pyrogens are heat-inducing molecules. That induces heat, so the inflamed area gets very hot. Because fluid is coming in rapidly, the area gets swollen. And as the tissue swells, that presses in on nerves and causes pain and loss of function. So that's why these signs of inflammation characterize what's going on here. But there's much more than just fluid going on here. And what we see in all inflamed sites is an accumulation of large numbers of inflammatory leukocytes, or inflammatory white blood cells. And these come in various waves. So this is not very clear, I apologize, but that word says neutrophils there. Neutrophils come in very early, followed by macrophages, then followed by T cells and mast cells. And these neutrophils and macrophages play essential roles in trying to fight any infectious agents that's got into the inflamed site. And they do this very effectively because they're armed with enzymes that can kill infectious agents really very, very efficiently. And not only that, but especially the macrophages are also involved in resolving inflammation. So all these signs of inflammation eventually switch off and a tissue settles back down to its normal homeostatic state, and macrophages are essential for that. And also in the context of inflammation that's arisen through wounding, macrophages are important for wound healing and various other aspects of this. And this wave of cells, this response to damage or infection that we call inflammation, is part of what we refer to as the innate immune response. So let's frame the key question that we're asking this morning, and that is, how does this happen precisely in time and space? So I could be bitten by an insect here, and I need to set up inflammation just here, not here or anywhere else. I can have inflammation in my lung here, and I don't want the inflammation to spread anywhere else. It must be localized. So here's a mosquito biting, and you'll set up inflammation precisely here, not anywhere else in the skin. Here's a wound. Inflammation localizes to the wound and not anywhere else. So how do we achieve that? And the conclusion from this is it's localized, therefore there must be some sort of local warning signal that rings out when damage occurs in precise areas that tells inflammatory cells where to go to reach inflamed sites. So let's now consider what this warning signal might be that goes off. And this is a silly analogy, but it's a useful one. Many houses, my own included, have set up burglar alarms. And when somebody breaks into our house, the burglar alarm goes off, and it actually will tell you which room the burglar has entered through. So that's an alarm system that tells us where and when somebody has entered our house, and it's a protection. And we have molecular alarms that do the same thing, and they're called toll-like receptors. And toll-like receptors respond to damage or infectious agents such as bacteria, viruses, and parasites. And as soon as any damage occurs at the precise area where damage occurs, these molecular alarms ring off and say, we have a problem here. We've either been damaged or breached or infected by infective agents. And that's what initiates the localized immune response, inflammatory response. So let's talk a little bit about toll-like receptors. Toll-like receptors were first identified in Drosophila, and Drosophila is a really elegant model for lots of early developmental processes that we still have as human beings, and the Drosophila has a very basic but useful innate immune response, and it can protect itself from infectious agents. And this fly here is a wild-type Drosophila, nice and healthy, looks absolutely fine. This poor insect here is a mutant Drosophila, and this was called a toll mutant many years ago when it's identified. This, of course, is the beauty of Drosophila, that it mutates rapidly, and so one can identify many mutations and characterize them and identify molecules involved in key biological functions. And this poor fly has a mutation in a toll gene, and that toll gene is a gene that rings off alarms when fungus gets into the Drosophila. This fly cannot ring that alarm off, and therefore the fungus gets in and can take over the fly and eventually kill it. So this led to the identification of toll-like receptors that are the alarms that trigger responses when any infectious agents enter our bodies or when our bodies are damaged or insulted in any sort of way. And Drosophila has one toll-like receptor. We have around about 10, and mice have about 12. And these respond to infectious agents and tissue damage by identifying molecules that characterize pathogens. So pathogen-associated molecular patterns are what we called. And there's an important concept here, and that is that in terms of our evolving interaction with pathogens, we're constantly playing a game of cat and mouse where the pathogen will evolve away molecules that we recognize, and then we'll try to re-recognize it. The key thing about the toll-like receptors is that they recognize molecules that the pathogens cannot evolve away. They're so fundamental to the function or structure of the pathogen that they can't get rid of them. So this is a slightly out of focus diagram. I apologize for that. But the sort of molecules that we recognize using toll-like receptors are cell wall components that the bacteria simply cannot get rid of, essential fungal components, flagellin, which is involved in bacterial moving, and in terms of viruses, double-stranded RNA, which of course we don't have in our cells, but the virus does. We recognize it as unusual and respond to it. Odd single-stranded viral RNA, and certain bacterial types of DNA. So these toll-like receptors have evolved over hundreds of millions of years to identify molecules that are unique to the pathogens and that the pathogens simply can't live without. So we ring out this alarm as soon as we see these. And these molecules also respond to damage that's not infected through damage-associated molecular patterns, which are breakdown products of our own proteins. So we now have this alarm system in place that tells us precisely where in space and time an infection or a damage has occurred. And these receptors are expressed in immune and inflammatory cells, but also importantly on epithelial and lots of other cells, particularly cells at the border between us and the outside world, like the skin and the lung and the gut. And they have a conserved molecular structure. They're transmembrane proteins in the main, although some sit within the cell, and the extracellular part responds to the pathogen-associated molecular pattern or the damage-associated molecular patterns and fire in a signal that starts to trigger the inflammatory response. So how do toll-like receptors actually trigger an inflammatory response? Well, what they do, and I have a real scientific dislike of cellular signaling. It's just a personal bias, so you're not going to get much cellular signaling. But essentially, toll-like receptors signal through a couple of pathways, the outcome of which is the induction of interferon expression and inflammatory cytokine expression. What that then does is to set up localized inflammatory cytokine-driven responses at the precise area of damage. You don't ring out any signals here, so this remains okay, it's not inflamed. You do ring out the alarm here through the toll-like receptors, so this starts to get inflamed because you're making these inflammatory cytokines and interferons. Now that's okay, but that does not tell us how you manage to get inflammatory cells to these specific sites. It's great to set up inflammation with cytokines and interferons, but how does that help us get inflammatory cells right to this area here and very precisely? And the key to that is the real focus of this talk, and that's molecules called chemokines. Because once you make inflammatory cytokines and interferons, you also trigger the expression of molecules called chemokines, and these are the fundamental regulators of inflammatory and immune cell migration to specific places in space and time. And I have studied chemokines, sadly, for almost 30 years now, and these have been my scientific curiosity and passion for that length of time. So I want to try to persuade you, of course, that these are the most important molecules in the world. So let's now think about chemokines, and we'll try to build up a picture of how chemokines regulate the inflammatory response and all the different ways that we believe that they might do that. Let's start with a really important definition. What are chemokines? Well, the word chemokines is an amalgamation of two words, chemotactic cytokines. These are cytokines that induce chemotaxis. And chemotaxis is the process of directional cellular migration. So what chemokines or chemotactic cytokines do is they induce the directional migration of inflammatory and immune cells and other cell types that we'll touch on a little later on. So that's their fundamental property. But I want to really make the definition of chemokines very precise. There are many cytokines that are chemotactic, but that are not chemokines. So for example, transforming growth factor beta, or TGF-beta, which some of you may have heard about, an extremely important molecule in development and in adult biology, is chemotactic, and it's a cytokine, but it is not a chemokine. Interleukin-2 and interleukin-18 are also chemotactic, and they're cytokines, but they are not chemokines. So how do we precisely define a chemokine? We define it not simply on the basis of the fact that it can be chemotactic, but most importantly, on the basis of its biochemistry. So chemokines are defined by a conserved cysteine motif. And I don't know how much protein chemistry you know, but just to put this in context, this is a line diagram of a chemokine. We always draw proteins in the amino terminal to carboxy terminal direction. And the C's are cysteine amino acids. These black lines here just represent other stretched amino acids, which are not conserved, but these cysteines are conserved throughout the chemokine family. So the presence of a cysteine motif like this, and I'll tell you in a second why this is important, is a defining feature of the chemokines. And without one of these cysteine motifs, you can be as chemotactic as you like, but you're not a chemokine. So this is the defining feature. Now, we subdivide the chemokine family, and it's a big family. We as humans have around about 45 different chemokines. We subdivide this large family into 4 subfamilies according to the specific distribution of cysteines. So this first subfamily has 4 cysteines. The first 2 are right next to each other. This is called the CC chemokine family. This is the CXC chemokine family, and X represents a single variable amino acid between the first 2 cysteines. And this again has 4 positionally conserved cysteines. This chemokine family is only 2 cysteines. This is the XC family, and this chemokine family has 4 cysteines, but with 3 amino acids between the first 2 cysteines, and it's called the CX3C family. And we name chemokines according to this definition. So this chemokine subfamily is called the CC chemokine family. CC ligands, L for ligands, so CCLs. And the numbering system works in terms of the numbers in which the genes were deposited in the databases. So CCL1 was the first CC chemokine identified, and CCL28 the last. We probably know all the chemokines now. There are no surprises out there. CXC chemokines are called CXCL ligands. 1 was the first one, 17 the last one, and these are much smaller subfamilies, XCL1 and CX3CL1. So the chemokines are the major regulators of chemotaxis of leukocytes. And the next question then that relates to this is, why are these cysteines so important? They've been conserved throughout evolution, so they must play some sort of important role. And again, a bit of protein chemistry for you, which you may or may not know, will help explain why these are important. Basic amino acid structures like this. You have an amino group and a carboxy group, and you build up proteins in this direction, which is why we always refer to proteins in amino terminal to carboxy terminal direction. And amino acids add on in this direction through formation of what's called a peptide bond. Now we have 28 different amino acids, and what makes them different is the fact that they have different side chains. And this gives amino acids different biochemical properties. And cysteines have a side chain called a sulfhydryl side chain, which is a sulfur and a hydrogen. And in secreted proteins, when these become close, they become oxidized and they form what is called a disulfide bond. And that is a covalent bond, that is a strong chemical bond that cannot be broken down. And these are important because what they do is they pin structures together. These yellow lines are the cysteine bonds in chemokines. So they not only help the protein to fold, but they lock it together. And that's another important point about proteins, is we very typically draw them out in a linear fashion, but what proteins do is that this line, this chain of amino acids, twists and folds into beautiful, perfectly precise structures that are essential for function. And in the chemokines, these disulfide bonds are essential for pinning this structure together, otherwise it falls to pieces, and that is why they've been so highly conserved. So that is why cysteines are conserved throughout the chemokine family. So where did chemokines come from in terms of evolution? The first, probably almost certainly the first chemokine that evolved was a molecule called CXCL12 around about 600 million years ago, probably, with early fish. And CXCL12 deficient mice die around about birth for reasons that I'm going to tell you about in the next slide. So they're a vertebrate invention. Drosophila have no chemokines. They're only found in vertebrates, but they go back about 600 or 700 million years. A lot of very elegant studies in chemokines have been done using zebrafish. These are fish that you can buy in pet shops very easily, and they're a nice, elegant model for early vertebrate development. So we know a lot about the primary roles for chemokines by studying fish. What these have told us is that this ancient chemokine, CXCL12, the name's not important, don't worry about it. It's just, again, a CXCL chemokine ligand, the 12th to be identified, that's why it's called that. Its main role, its ancient role, was to regulate stem cell migration during development. And so what it does is it regulates the movement of hematopoietic stem cells in the embryo. For us, where hematopoiesis sits mainly in the bone marrow, it is essential for stem cells navigating to the bone marrow. Any of you who have ever had or know somebody who's had a stem cell transplantation, this axis involving CXCL12 and its receptor is essential for getting stem cells to the bone marrow. And if you knock this out or interfere with it, you disrupt bone marrow hematopoiesis, which is fatal. So stem cell migration was the key primordial or ancient role for chemokines. There's another really important developmental stem cell type called primordial germ cells, and these are involved early in development in the embryo, and these are the precursors for sperm and eggs. So these are the germ cells that give rise to our species, basically. And in development, what the primordial germ cells do is they navigate to an area called the genital ridge, and they need to go there to develop properly. And this axis is regulated by this ancient chemokine, CXCL12. And if you knock it out, any species that are born, including zebrafish, are sterile because they cannot make sex cells, sperm and eggs. And also neural progenitors and vascular progenitors are dependent on CXCL12 for their proper navigation in the embryo. So the ancient role for this first chemokine, CXCL12, was to regulate stem cell migration in the embryo. And over the 600 or 700 million years since then, the family has got much, much more complicated through simple gene duplication, etc. And we have around about 45 chemokines now that involve themselves in sometimes extremely subtle ways in regulating immune and inflammatory cell migration. Incidentally, some fish have around about 150 chemokines, so we are not the most complex in terms of chemokines. But it is complicated. And if you leave here with no other message other than that chemokine biology is inherently complex, then I've achieved at least something. So we tend to cheat in the chemokine field, and we simplify the biology by defining chemokines, broadly speaking, as being either homeostatic, that is, they're involved in basal trafficking of leukocytes in health and disease. So I said earlier that I have T cells in my skin, and my skin is not inflamed or in problem. They just go there because it's a homeostatic role. And the stem cell migration is homeostatic, and that's one of the roles. The other role that we're going to look at today is a role for chemokines in regulating the migration of these really important inflammatory cells. So regulation of inflammatory cell migration. So broadly speaking, we define chemokines as either being inflammatory or homeostatic according to the in vivo context in which they function. Let's think about inflammatory chemokines now. Inflammatory chemokines are not expressed in healthy tissue. This is healthy skin here. As soon as the tissue is damaged or infected or insulted in any way, we make extremely high levels of chemokines in any cell type that's experiencing damage, any cell type in the body. These recruit inflammatory cells such as neutrophils, monocytes, and mast cells. Many are made at the same time. Once they've done this, they then switch off, and the whole process resolves itself. So that is how we get tissue-specific accumulation of inflammatory cells to sites only of damage or infection, because only here do we ring off the toll-like receptor signal, and only here do we make the inflammatory chemokines that induce the cell types that trigger the inflammatory response. I hope that makes some sense, but we'll summarize this at the end of the talk as well for you. So the next question is, chemokines are secreted proteins. They attract inflammatory cells at the sites of damage, but how do they do that? How do the inflammatory cells detect the chemokines? How do they sense them? And they do that because chemokines bind to receptors present on inflammatory cells, which are 7 transmembrane spanning receptors. And so that you know something about this family, these are typically coupled to G proteins for signaling. And we have receptors for CC chemokines. We have 10 of these so far. Receptors for CXC chemokines, and receptors for the different subfamilies of chemokines. So this is a very well characterized receptor family, and these receptors are distributed in various immune inflammatory cells. Some viruses have also picked up chemokine receptors from their host cells, and they use these in ways that we don't have time to go into to help with the process of infectivity in the viral life cycle. Now, in terms of inflammation, chemokine receptor biology is actually extremely complicated. We typically think of a ligand and a receptor having a one-on-one relationship, monogamous relationship. And this breaks down in inflammation. And we define inflammatory chemokine receptors, here's one here, CCR5, as being promiscuous in that they bind multiple ligands or chemokines. And the ligands are unfaithful to the receptors in that they don't just bind to one receptor, they bind to multiple receptors. And this makes the biology hugely complicated. So this receptor here, this is a CC receptor, the 5th one to be identified, hence CC receptor 5. This is an intensely studied receptor. It is the key receptor involved in HIV pathogenesis, a major drug target. It is promiscuous in that it binds at least 4 chemokines, probably 8 to 10 chemokines with high affinity. And each of the chemokines it binds is unfaithful to it. So this one here, CCL3, does not just bind to CCR5, it also binds to CCR1, CCR3 in a molecule that we work with called ACKR2. And that complicates the biology to a huge extent, meaning that really, if we're totally honest, we don't fully understand how we properly orchestrate inflammatory responses using chemokines. We know they're involved, but we don't really understand all the details yet. So again, a key take-home message is that we don't really understand this process yet, and it's okay to say that. Again, we define receptors as being either inflammatory or homeostatic. These are the key receptors involved in getting inflammatory cells to inflamed sites, and these are involved in trafficking of cells in health homeostatically. And this property of promiscuity and unfaithfulness is largely associated with the inflammatory receptors in green here and not with the homeostatic receptors. And the idea is that the ability of multiple receptors to respond to multiple chemokines and multiple chemokines to interact with lots of receptors gives great scope, versatility and redundancy to inflammation. That's the idea I've written probably there because again, we simply don't know the answer to that. People will tell you that's the case, but we simply do not know it. It's not clear and a lot of work has to be done to try to define this. So let's go back to the title, and the title was about the inflammation highway. So chemokines are the signposts on the highway. This is a circulation here. Here's cells zipping along the circulation. And when you trigger damage through toll-like receptors and you switch on the chemokines, that triggers up an exit signal saying, come off here, please. This is where the damage is. We need to get inflammatory cells in here. But the next question is, how do we do this? Where do cells actually see chemokines? And they see chemokines on the inside of blood vessels. So this is something that you may have seen. This is the famous leukocyte adhesion cascade. This is the inside of a blood vessel. These are endothelial cells lining the blood vessel. This is called the luminal face of the endothelial cells. This is the subluminal face. And when you get inflammation through infection or tissue damage, you trigger signaling here. This induces chemokines. The chemokines are presented here on this face and they start to attract the chemokines. So that is the exit sign here on the luminal face of the blood vessels. Now there's a problem with this model, and that is that blood flows, and blood flows extremely rapidly. It's been worked out. This is this beautiful fountain from Lake Geneva that if you put your finger at the nozzle here, you would feel the pressure that you have in blood in an average blood vessel. So won't the chemokines get washed away? And just to cut a long story short, and there's a lady listening to this talk who is the world expert in this, it's Amanda Proudfoot. Hi, Amanda. They're held on to the surface endothelial cells in what's called the glycocalyx, which is sort of sugary coating on top of the cells, by binding to molecules called glycosaminoglycans. And these are sugary molecules that hang on to chemokines and hold them and stop them being washed away by the force of the blood. And that therefore allows the signal, this exit signal, to be held here, so cells roll along, they see the signal, and they know precisely this is the time to exit the tissue and move into the inflamed tissue. That's how they know where they are in space and time. It's chemokines presented in the blood cells that tell them where to leave, just like an exit sign from a motorway. So again, just to cut a long story short, when you're coming off the motorway, you put the brakes on and you head off. So how do you put the brakes on in the cells here? Because these are really moving along. How do you stop them physically? You stop them because what chemokines do is they signal into the cells and trigger integrins, which allow firm adhesion. So these cells now lock down, the brakes are on, and they can start to move into the tissue. And we're not going to go into this because we don't have time to talk about this. But again, this involves what's called inside-out signaling. The chemokine signaling into the cell, the inflammatory leukocyte, triggering activation of integrins, which then lock down onto the surface and allow the cell to lock on and exit. So let's put all this together in the last couple. I hope I've got a couple of minutes left. What happens is that at a precise point where you get infection, damage, or wounding, or any sort of insult to the tissue, you set off local alarm signals through the toll-like receptors. These lead to the production of localized chemokines. These are the chemotactic molecules involved in attracting these cells to the damaged sites. This leads to site-specific inflammatory cell recruitment, and is fundamental to how we let cells navigate on this inflammation highway. Now, just a final point is that there are profound therapeutic implications of this, because whilst we can get specific recruitment of cells to damaged sites, we can also get inappropriate recruitment of inflammatory cells to normal sites that lead to really debilitating diseases such as rheumatoid arthritis, psoriasis, multiple sclerosis, and damage in a whole range of different pathologies. So that makes inflammatory chemokines and their receptors fundamentally important targets for therapeutic implications. But really, as a field, we failed dismally to capitalize on this. And that's part of the reason, part of the reason for this is what I said earlier, that we really don't understand this family well enough yet. But this is a key goal for the future, is to try to therapeutically interfere with chemokines and receptors. So that's all I want to tell you. I hope that was clear. If it's not, I believe we've got some time for questions now, but email me as well and ask questions. That is how blood cells know where they're going in space and time. Chemokines are the fundamental regulators of that process, and that is how we set up localized inflammatory responses. So thank you for your attention, and I'd be delighted to take any questions, and I hope my Scottish accent wasn't too much for you. Thank you.
Thank you very much, Jerry. That was a great presentation, actually. So I have a couple of questions, and as chair of the session, I'm going to take my prerogative and actually ask those questions. So you mentioned that CXCL12 is absolutely important in terms of reproduction. So my question to you is there any redundancy in the context of CXCL12 in the chemokine system?
No, that's a great question. This idea of redundancy relates to inflammation. When you start to get down to chemokines receptors involved in important developmental and adult processes, there's no redundancy. There is no backup system for CXCL12, and if one deletes it in mice or fish, there are profound problems. There's no redundancy there, so that the mice are deeply sick. The fish do survive, but they're infertile. They have a messed up immune and brain system, so there's no redundancy there at all. That's a fundamental regulator that is so important that if it goes wrong at all, it's not compatible with survival beyond birth.
So that's remarkable. So I have a sort of follow-up question to that, because you also mentioned that different chemokines bind to the same receptor. So is this redundancy? In other words, can we replace one chemokine with another? Or do they bind differently to the same receptor and result in different responses? And if that is the case, are there any examples you can cite for us?
Yeah, again, a great question. As the biochemistry and the pharmacological understanding of chemokine receptors develops, we become aware of this thing called biased signaling. And biased signaling is something that the G protein coupled receptor people love. And the idea behind that is that multiple ligands for the same receptor can actually interact in subtly different ways with that receptor and bring about different downstream signals. So whilst we talk about redundancy, we don't really know that redundancy is what's going on here. There might be exquisite subtlety and complexity. You know, either way, it's hard to understand. If you inflame a tissue, you'll make 20 different inflammatory chemokines at the same time. What are they doing? Are they redundant? If they're subtle, then heaven knows how you work out what on earth is going on. So these are wide open questions, Eleanor.
All right, so I've got a couple of questions in our chat room.
Thank you.
One from your colleague Amanda Proudfoot.
Hi, Amanda.
Who says, CXCL12 exists in many isoforms. Which are the most important, or has this not been delineated?
So CXCL12 has a number of splice isoforms which have different carboxy termini. That's the far end of the chemokine, the right-hand side of the chemokines. One or two reports suggest that these are important. The main one is CXCL12 alpha, but they're sporadic reports, and it's not clear that there's fundamental differences. There certainly are reports, but there's not a consistent literature out there that tells us what these different isoforms are doing. Zebrafish have two isoforms of the receptor for CXCL12, and they do different things, but fish are slightly unusual in terms of genetics. So in terms of mammals, we have isoforms, but we're not entirely clear yet what their relevance is.
All right. We have another question here. Can you please comment on whether shift in microbiota as a result of antibiotic use can change the local expression of chemokines in distant tissues?
Great, that's a great question. Nobody's looked at it, but put it this way, it would not surprise me. The gut microbiota has profound effects on distant tissues. It totally dictates the outcome of Th17 biology. So nobody's looked at it, but heaven knows I would expect that to be the case. And you also have microbiota in the lung, and that's got to have an effect on the local receptor expression in cell types. So it's a wide open research question for somebody to look at, but it would I would be surprised if it didn't. It affects almost everything else, which of course gives us a whole bunch of other problems.
Then another question you alluded to about viruses, but somebody here is asking, how do some viruses like bacteriophage escape chemokines in the body?
Oh, right. It depends on the virus. Some viruses come and go so quickly it's not an issue, but some viruses are fascinating things. I mentioned earlier in the talk that some viruses have got chemokine receptors in their genome. And what DNA viruses do is they insert in the host genome, and when they come out of the host genome, they sometimes take host genes with them. And if they can use them, they will, and if they can't, they get rid of them. They do it extremely quickly and efficiently. So if a virus has a gene in its genome, it's there for a reason. And viruses have also picked up genes that they have used to neutralize chemokine function. So a number of viruses, when they infect, will also express proteins that block chemokine function, and that's one of the ways that they can evade immune and inflammatory responses. A number of other pathogens, our colleague Amanda Proudfoot has studied this in ticks, and they do the same thing. So there's a common theme here. And this also has important implications for therapy, that viruses and ticks have found ways of blocking chemokine function so that they can interfere with the host without being rejected. And we need to try to learn from them and work out ways of doing this therapeutically in inflammatory disease. It's a great question.
So perhaps the last question here before we move on is from Keith Dadson, who first of all comments on it as a great talk, a wonderful talk. And he asked, what is known about chemokines and chronic inflammation, or the inability of a tissue to turn off the inflammatory response?
So again, it's a great and complex question. If you look in rheumatoid joints, for example, you'll find every chemokine you want there. The issue is, I think, more what role they have in the initiation of the process. That's the interesting one, the things are building up. Once you start to get damage, that damage will amplify the inflammatory response and the chemokine response, and what you get is a whole mess which combines the involvement of chemokines in the start of the pathology with also its response to the damage caused by the pathology. So if you look in any inflammatory or immune pathology, you can find any inflammatory chemokine you want. They're stuffed full of neutrophils, macrophages, mast cells, inflammatory T cells. So it's a whole mess. And again, that's why some sort of broad-based ability to interfere with them, the way the viruses have done, will be important. And if you look in the rheumatoid literature, there's a whole bunch of literature about chemokine receptors, but again, we have failed miserably to tackle it because it's just so damn complicated. But a great question, thank you.
Well, all that remains now is for me to thank you, Jerry, for a great opening talk. And
thank you very much. Thank you.
I know there'll be a lot of folks around the world now who are very much more informed and up to date. So thank you very much.
And please email me if you have any questions. Thank you very much.